Hello everyone, I Pooja Verma welcome you all in the second session of chapter 12, that is mineral nutrition. In this session we will discuss about deficiency symptoms of essential elements, toxicity of micronutrients, mechanism of absorption of elements, nitrogen cycle, biological nitrogen fixation and fate of ammonia molecule. Learning objectives of this session are to help you learn about deficiency symptoms of essential elements, to help you understand the concept of toxicity of micronutrients, to help you learn about the mechanism of absorption of elements, to familiarize you with the nitrogen cycle in nature, to help you appreciate the process and importance of biological nitrogen fixation, to develop scientific attitude, creativity, aesthetic sense and problem solving approach, to enable you to find applications of these topics in your daily life. We have already discussed in the previous session that 16 elements are considered essential to plant growth. What will be the effect on plant growth if the supply of essential elements become limited? A deficiency of one or more of these essential elements leads to a reduction in growth, yield and crop quality. The concentration of essential elements below which plant growth is retarded is called critical concentration. So, plants must be supplied essential elements above critical concentration. In the absence of any particular element, plant will show morphological changes called deficiency symptoms. In which part of the plant deficiency symptoms will first appear depends upon whether the element is mobile or immobile. If the element is mobile, it means it can move quickly from one part of the plant to the other. In this case, deficiency symptoms will first appear in the older tissue. This happens because young tissues are actively growing. They require essential elements in large amount. Elements from the older part will get dissolved and move to the younger tissues. Thus, the requirement of younger tissues will be fulfilled, but older tissue will show deficiency symptoms. Examples of this category are nitrogen, potassium and magnesium. The opposite scenario will occur if the elements are relatively immobile. Young tissues, as they are actively dividing, require essential elements in large amount. But since the element is immobile, it cannot move from the older plant parts to the younger tissue. Thus, deficiency symptoms appear first in the young tissue. Example of this category is calcium. Now, let's discuss some of the common deficiency symptoms in plants. First, let's discuss about chlorosis. Chlorosis refers to the loss of normal green coloration of leaves of plants. In this condition, leaves produce insufficient chlorophyll, thus they turn yellow. You have been learning since your lower classes that chlorophyll is responsible for the green color of leaves. Thus, chlorotic leaves are pale yellow or yellow-white. The affected plant has little or no ability to manufacture food through photosynthesis, and unless the cause of its chlorophyll insufficiency is treated, the plant will die. Magnesium ion is present in the ring of chlorophyll molecule, so one might think that only deficiency of magnesium can develop chlorosis condition in plants. But deficiency of nitrogen, potassium, sulfur, iron, manganese, zinc and molybdenum can also develop chlorosis condition in plants. Second deficiency symptom is necrosis, in which death of tissues, particularly leaf tissues, occurs. The affected plant tissue usually turns brown to black in color. Necrotic symptoms could appear in any part of the plant, such as in leaves or in woody tissues. Necrosis is caused due to deficiency of calcium, magnesium, copper and potassium. In the absence or low level of nitrogen, potassium, sulfur and molybdenum, the process of cell division gets inhibited. Thus, plant growth is retarded. Inhibition of cell division is a major deficiency symptom for plants. Do you know delayed flowering is also a deficiency symptom? If the plant is deficient in nitrogen, sulfur and molybdenum, flowering will be delayed in plants, which is a major deficiency symptom. The correct diagnosis of nutritional deficiency in plants is important in maintaining optimum plant growth. Interpreting visual nutrient deficiency symptoms in plant can be difficult. So, plant analysis and soil nutrient testing is necessary to confirm nutrient stress. Plants require micronutrients in low amount. If plants obtain them in moderately less amount, this causes deficiency symptoms as we have already discussed. But in higher doses, micronutrients can become toxic. Thus, there is a narrow range of concentration in which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of tissues by 10% is considered toxic. Let's try to understand this with the help of an example. Manganese is toxic beyond 600 microgram per gram for soybean plants. If the plant is having toxicity of manganese ion, brown spots surrounded by chlorotic veins will appear on the leaves. Toxic effects may be due to the excess of micronutrients in the plant or due to its interference in the absorption and functioning of other nutrients. Manganese competes with iron and magnesium for uptake and with magnesium for binding with enzyme. Manganese also inhibit calcium translocation in shoot apex. Therefore, excess of manganese can cause deficiency of iron, magnesium and calcium in plants. Thus, in terms of manganese toxicity is actually caused by deficiency of iron, magnesium and calcium in plants. We know that plants also require elements for their proper growth and development. We will now discuss how plants absorb elements. Mineral salts are mostly present in the soil solution in ion forms. Plants absorb them in the form of ions. Plants use two methods for ion absorption. The first method is called apoplast pathway, in which there is a rapid uptake of ions into the outer free space of cells. In this pathway, water and ion molecules move from root hair to xylem through cell wall region of intervening cells. You have studied about plasmodesmata, which are microscopic channels that join the cytoplasm of two adjacent plant cells. In the second method called symplast pathway, water and ions move across the living symplast region, which consists of cytoplasm and plasmodesmata. The movement of ions into or out of the cells is called flux. You must be familiar with the term nitrogen cycle. Nitrogen is one of the major micronutrient needed by plant for proper growth, as nitrogen helps in the formation of amino acids, proteins, hormones, chlorophyll and many vitamins. The formula of nitrogen found in the atmosphere is N2, in which two nitrogen atoms are joined by triple bonds. Nitrogen is one of the most abundant gas in our atmosphere, as it constitutes about 78%. Molecular nitrogen is a highly inert gas. Thus, it is energetically difficult for most of the plants to use it directly. Therefore, nitrogen is a limiting nutrient for both natural and agricultural ecosystem. Plants must fix nitrogen to form compounds such as nitrates, nitrites, ammonium ions, etc. before it is absorbed and utilized by the plants. Such conversion of nitrogen into its different forms constitute the nitrogen cycle. It is a biogeochemical cycle by which nitrogen is converted into multiple chemical forms and circulates among atmosphere, terrestrial and marine ecosystem. Atmospheric nitrogen is converted into usable forms by various agencies of nitrogen fixation. The process of conversion of atmospheric nitrogen to ammonia is termed as nitrogen fixation. There are three methods of nitrogen fixation. The first one is the biological nitrogen fixation, the process in which atmospheric nitrogen is converted into usable forms of nitrogen by the agency of living organisms is called biological nitrogen fixation. Rhizobium bacteria, cyanobacteria and various other microorganisms help in the process. Farmers add urea in their fields to increase the yield of crop plants. Urea is an excellent source of nitrogen for the plants. It is manufactured in the factories. This process is called industrial nitrogen fixation, in which atmospheric nitrogen is converted into ammonia. This is the second method of nitrogen fixation. You might have heard people often say that rainwater is good for growth of plants. Have you ever wondered why is it so? It is because during the process of lightning, in the clouds, atmospheric nitrogen N2 reacts with oxygen to form nitric oxide. The nitric oxide NO is again oxidized with oxygen to form nitrogen peroxide NO2. The nitrogen peroxide can combine with water to form nitrous acid and nitric acid, which fall on the ground along with rainwater and reacts with alkaline radicals to produce water-soluble nitrates and nitrites, which are directly absorbed by the plants. It acts as a rich source of nitrogen and helps in the plant growth. This constitutes the third method of nitrogen fixation called electrical nitrogen fixation. Nearly 10% of nitrogen fixation occurs by this process. Pseudomonas and thiobacillus bacteria present in the soil convert soil nitrate ions into atmospheric nitrogen. This process is called denitrification. It is a wasteful process in which fixed nitrogen is again returned back to the atmosphere. As you can see nitrogen cycle on the screen, biological nitrogen fixation and industrial nitrogen fixation convert atmospheric nitrogen into ammonia. During electrical nitrogen fixation, atmospheric nitrogen is converted into nitrate ions. Ammonia formed by biological and industrial nitrogen fixation is converted into nitrite ions by nitrosomonas and nitrococcus bacteria. Nitrite ion is converted into nitrate ion by nitrobacter bacteria. These nitrifying bacteria are chemoautotrophs as they prepare their food by utilizing the energy released during the chemical reaction catalyzed by them. They are not dependent upon the solar energy to prepare their food. These steps are called nitrification. Nitrite and nitrate ions present in the soil are absorbed by the plants. These ions are absorbed by the green plants and transported to leaves, reduced to ammonia that finally forms amine group of amino acids. Plants then use this ammonia for making various biomolecules like proteins, vitamins and nucleic acids. Once these plants are consumed by animals, this nitrogen becomes part of animal biomass. And once these plants and animals die, organic nitrogen present in them in the form of proteins, vitamins and nucleic acid is again converted into ammonia. This process is called ammonification. Thus, the nitrogen cycle continues. We have already discussed about biological nitrogen fixation in the nitrogen cycle, the process in which reduction of nitrogen into ammonia takes place by living organism is called biological nitrogen fixation. Enzyme nitrogenase, which helps in the process of nitrogen reduction, is found exclusively in prokaryotes. Those microbes which contain this enzyme are called nitrogen fixers. You can see the reaction catalyzed by nitrogenase enzyme on the screen. We can classify nitrogen fixing microbes into two categories such as free living or symbiotic. You have studied about rhizobium bacteria in your lower classes. A rhizobium bacterium shows symbiotic relationship with leguminous plants and is found inside their roots. Azotobacter is a free-living nitrogen fixer commonly used by farmers while growing maize. In chapter 2, you have studied about cyanobacteria such as Anabaena and Nostoc, which helps in the process of nitrogen fixation in paddy fields. They are also free-living nitrogen fixers. If a leguminous plant is growing in or around your home, uproot any one plant of a common pulse just before the flowering. Observe the roots carefully. You will notice prominent outgrowth on the roots which are called root nodules. With the help of a knife, carefully cut these root nodules. You will notice that the central portion of these root nodules is reddish or pinkish in color. Why such root nodules are pink? It is due to the presence of a pigment called leguminous hemoglobin or simply leg hemoglobin. It is an oxygen-carrying pigment found in the root nodules of leguminous plants. Leg hemoglobin has 10 times more affinity for oxygen as compared to the blood pigment hemoglobin. It acts as an oxygen scavenger as the enzyme nitrogenase is highly sensitive to molecular oxygen. This enzyme requires anaerobic condition for its proper functioning. Leg hemoglobin pigment present in the root nodules protects nitrogenase enzyme from oxygen. Let's now discuss the various steps through which root nodules are formed in the roots of leguminous plants. Several species of rhizobium bacteria live in the soil. They are unable to fix nitrogen by themselves. In order to fix nitrogen, they must enter the roots of leguminous plants. Rhizobia multiply and colonize the surroundings of roots and get attached to root hair cells. Roots of a leguminous plant secrete several chemical attractants like flavonoids and betaines. Rhizobia collect over the root hairs curl and bacteria enter inside the root hair. An infection thread is produced which carries bacteria into the cortex of the root. The infection thread grows along with the multiplication of bacteria. The bacteria are then released from the thread into the cortical cells. The infected cortical cells differentiate and start dividing and produce nodules. Nodules thus formed establish a direct vascular connection with the host plant for exchange of nutrients. You can see all the steps of nodule formation on the screen. Nodule provides all the necessary biochemical components required for nitrogen fixation, such as the enzyme nitrogenase, leg hemoglobin, and reducing molecules like NADH. Nitrogenase enzyme has iron and molybdenum. Both of them take part in attachment of a molecule of atmospheric nitrogen N2. Bonds between the two atoms of nitrogen become weakened by their attachment to the metallic components. The weakened molecule of nitrogen is acted upon by hydrogen. The first hydrogen molecule is added. Due to reduction, triple bond gets converted into double bond. Then the second hydrogen molecule gets attached to the enzyme nitrogen complex. This further reduces double bond of nitrogen into single bond. Once the last and third molecule of hydrogen gets attached, it is converted into ammonia. Nitrogenase requires 8 molecules of ATP for each ammonia molecule produced. You can see the process and reaction on the screen. Ammonia is the first stable product of nitrogen fixation. It is toxic even in small quantities. At physiological pH, ammonia is protonated to form ammonium ions. Plants utilize these ammonium ions to synthesize amino acids by two processes. First process is called reductive amination. Reduction means addition of hydrogen. Amination means addition of amino group. Alpha ketoglutaric acid is reduced, means hydrogen joins with it as well as amino group is attached to it in the presence of ammonium ion, NADPH and glutamate dehydrogenase enzyme to form glutamate. You can see the reaction on the screen. Second process is called transamination. It involves the transfer of amino group from one amino acid to the keto group of a keto acid. Glutamic acid is the primary amino acid involved in transfer of amino group to as many as 17 amino acids. The enzyme transaminase catalyzes the reactions. Alpha ketoglutaric acid is formed again, which can be reused in the first process. You can see the reaction on screen. In the last step, amides are formed by the plants. Amides are amino acid derivatives in which component of carboxylic group COOH is replaced by another amino group. Amides are double aminated keto acids. Glutamine and asparagines are the two most common amides formed by the amination of glutamic acid and aspartic acid respectively. Amides help the plant to store excess nitrogen and they can be easily transported within the plant. Let's now have a quick recap of what we have learned. In this session, we have learned about deficiency symptoms of essential elements, toxicity of micronutrients. We also discussed about the mechanism of absorption of elements by plants. We have also learned about the process of nitrogen cycle and the process of biological nitrogen fixation in great detail. Before we conclude, here is an assignment for you. Try to answer the following questions. Why rainwater is considered good for plant growth? Collect information on harmful effects of excessive fertilizers on soil. Click or collect 2 photographs of plants showing any deficiency symptom. What are biofertilizers and how are they helpful to the farmers? Learners, hope you all enjoyed the session. Keep smiling, keep learning. Thank you.